COVID-19 pandemic brought vaccine development into global consciousness in a way that no public health event had done in living memory. In a matter of months, billions of people who had never thought much about how vaccines work were suddenly following clinical trial phases, debating mRNA technology, and tracking emergency use authorization timelines with an intensity usually reserved for election results. The development of effective COVID-19 vaccines in under a year, compared to the typical decade-long timeline for a new vaccine, was a genuine triumph of modern science, made possible by decades of prior research, massive coordinated funding, and several fortunate features of the coronavirus itself that made it a tractable target. But it also laid bare the fragility of the traditional vaccine development pipeline and the enormous cost, in both money and human lives, of delays in that pipeline. Any technology that could meaningfully accelerate vaccine development deserves serious scrutiny. And quantum computing, for reasons grounded in the same molecular simulation science we explored in our drug discovery video, is one of the technologies that serious researchers believe may eventually play a meaningful role. Today we are going to examine exactly where quantum computing might intersect with vaccine development, how close that intersection actually is, and where the honest boundaries of current capability and future promise lie. To understand where quantum computing might help, you first need to understand what vaccine development actually involves at the molecular level, because the challenges are more specific and more interesting than the general phrase vaccine development suggests. A vaccine works by training the immune system to recognize and respond to a pathogen, a virus or bacterium, without causing the disease itself. The most fundamental challenge in designing a vaccine is identifying which part of the pathogen to show the immune system, how to present it, and how to ensure that the immune response generated will be effective against the actual pathogen when the vaccinated person later encounters it. These questions involve understanding the three-dimensional structure of proteins, the molecular machines that viruses use to enter cells, and that the immune system uses to recognize and attack pathogens, with a level of precision that determines the difference between a vaccine that works and one that does not. The COVID-19 vaccines based on mRNA technology work by instructing the body's own cells to produce a specific viral protein, the spike protein that SARS-CoV-2 uses to attach to and enter human cells, which the immune system then learns to recognize and attack. The success of this approach depended critically on understanding the three-dimensional structure of the spike protein in its active conformation, which in turn depended on structural biology techniques including cryo-electron microscopy and X-ray crystallography, combined with computational modeling. The ability to predict and understand protein structures has been one of the central challenges of molecular biology for decades, and progress in this area directly enables vaccine design. This is where quantum computing enters the picture, and where the connection to our earlier video on drug discovery is most direct. Many of the same molecular simulation challenges that limit drug discovery also limit vaccine antigen design. Understanding exactly how a viral protein folds into its three-dimensional structure, how it interacts with human cell receptors, how antibodies bind to it, and how mutations in the viral protein, the kind that produce new variants, affect all of these interactions, all require quantum mechanical calculations that become exponentially hard for classical computers. As the molecules involved grow larger and more complex, the quantum mechanical details of protein-protein interactions, electron distributions in binding sites, and the energetics of molecular conformational changes are precisely the kinds of calculations that quantum computers are theoretically well suited to perform more accurately than classical methods. One specific area where quantum computing might accelerate vaccine development is in the design of antigens, the molecular components of a vaccine that trigger an immune response. An ideal antigen is stable, easily recognized by the immune system, capable of generating broadly protective immunity, and manufacturable at scale. Designing antigens that optimize all of these properties simultaneously is an extraordinarily complex multi-objective optimization problem. Classical computational methods, including the increasingly powerful AI tools that have transformed structural biology, most notably DeepMind's AlphaFold system, have made remarkable progress in predicting protein structures from amino acid sequences. But predicting how subtle changes to an antigen structure will affect its immunogenicity, its stability, and its cross-reactivity with different viral strains involves quantum mechanical details that are beyond the reliable reach of classical approximation methods for complex molecular systems. A second area is in understanding viral evolution and predicting which mutations are most likely to produce vaccine-resistant variants. This is a problem with enormous public health implications. The ability to anticipate the emergence of new variants and preemptively design vaccines against them would transform the world's ability to respond to pandemics. predicting which mutations are energetically favorable for a virus, meaning which changes to its proteins are likely to emerge under evolutionary pressure, requires understanding the free energy landscape of protein folding and protein-protein interactions, which again involves quantum mechanical calculations that classical computers handle with approximations that may not be reliable enough for this level of predictive precision. A third area is in understanding immune system responses at the molecular level. The immune system's recognition of antigens involves extraordinarily complex molecular machinery: the binding of antibodies to antigens, the activation of T cells, the cascade of signaling molecules that coordinate an immune response, all of which involve quantum mechanical interactions at the molecular level. Understanding these interactions in sufficient detail to design vaccines that generate optimal immune responses, particularly for difficult targets like HIV or influenza, where broadly neutralizing immunity has proven elusive, may require quantum simulation capabilities beyond what classical computers can reliably provide. Now, as with every other application of quantum computing we have examined in this series, the honest accounting requires acknowledging the very large gap between theoretical promise and current practical capability. The quantum computers that exist today cannot simulate the molecular systems relevant to vaccine development with greater accuracy than the best classical methods. The molecular systems involved, viral proteins, antibodies, receptor complexes, are large enough and complex enough that they would require quantum processors with hundreds to thousands of error-corrected logical qubits to simulate accurately, far beyond what current hardware provides. The same fundamental hardware gap that limits quantum computing's near-term contribution to drug discovery limits its contribution to vaccine development, because the underlying scientific challenge is essentially the same: accurate quantum mechanical simulation of biologically relevant molecular systems. It is also worth acknowledging that the classical tools available for vaccine development have been improving at a remarkable rate and are already producing significant practical results. AlphaFold's protein structure predictions have been used in vaccine research and have accelerated the identification of antigen targets in multiple infectious diseases. Machine learning approaches to predicting immunogenicity, designing stable protein antigens, and modeling immune responses have made genuine progress. Quantum computing will need to demonstrate advantages over these increasingly powerful classical and AI-augmented methods, not just over older classical approaches, to earn its place in the vaccine development toolkit. The honest and hopeful picture is this: the COVID-19 pandemic demonstrated with terrible clarity what the world loses when vaccine development is slow, and it also demonstrated how much can be achieved when scientific resources are marshaled with urgency and focus. Quantum computing represents a potential future tool for understanding the molecular machinery of immunity and infection at a level of precision that current methods cannot match, with implications not just for COVID-19 but for influenza, HIV, malaria, tuberculosis, and the pathogens we have not yet encountered. That potential is grounded in real science, in the same quantum mechanical nature of molecular interactions that makes quantum simulation naturally suited to these problems. Realizing it requires quantum hardware to mature substantially beyond where it is today, and that maturation is happening on a timeline that is uncertain but that the scientific community is actively working to compress. Vaccine development has always been one of humanity's most powerful tools against infectious disease. Quantum computing may one day make it a sharper one.